Okay, welcome to today's Phenomics webinars, our first Phenomics webinar in the new year 2022. And I'm very happy to welcome you back here at our web space today with Dr. Arti Singh from Iowa State University, and she will be giving a webinar on image-based plan stress phenotyping in the fast-changing world of AI. A very relevant, a very recent topic that is certainly will continue to stay relevant throughout the next years and possibly decades. So why this is going to be, Arti will tell us today, at least a little bit of it, so much as you can tell for a one hour webinar. Before we dive into the world of AI, I wanted to quickly hint you guys at the upcoming International Plant Phenotyping Symposium that will be happening in late September 22. It's still a little bit of time. However, I wanted to use the chance to tell you that our website is online already from end of last year onwards. So there you can grab a few details as the planning progresses, such as information on the venue, information on the Wageningen NPAC facilities, as well as, of course, information about registration rates. And the abstract submission will open this week, a little bit later from today, and you can visit our website at www.ipps7.org. If you plan to attend this meeting, we are looking to have this meeting on site and in person. However, a few parts of the content will also be made available online at a later stage. And if you want to already have a look on the scientific sessions and topics, they are also displayed on our website, but you can see it from here. Also, this is particularly interesting for our speaker today, possibly a session on informatics, data, and analytics. So, yeah. Use the chance, visit our website. Information will be added there as the planning continues, and you are very welcome to send me an email at ippn@plant-phenotyping.org if you have any questions whatsoever. So, coming back at today's webinar, I want to use the time to quickly introduce Dr. Arti Singh, who is an assistant professor at Iowa State University. Arti Singh did her PhD in India and then continued to work in Canada. She has published two different books, one on disease and insect resistance in plants and another one on plant breeding and cultivated development. And she has been awarded a prestigious Borlong Women in Triticum Early Career Award in 2013. Back then she was already working in the field of machine learning and deep learning for plant stress phenotyping, and I'm very happy to welcome her here today. Hello, Arti.
Hi, Philip. How are you?
I'm very well, Arti, and glad that you are here with us today.
I'm
Sorry.
I'm very thankful for the kind invitation, Philip. Yeah.
It's our pleasure. Arti, one question. Where in India did you make your PhD?
So I did my PhD from GB Pant University, which is in North India, foothills of Himalayas.
A very nice region, as I have heard.
Yeah, it's a very fertile region. It's known as Rice Bowl of India, and yeah, we grow a lot of crops there.
Very, very good. So, Arti, I don't want to take up too much of the time. You are already here with your slides, and I will mute myself and listen to your webinar, Image-Based Plant Stress Phenotyping in the Fast-Changing World of AI. The stage is yours, Arti.
Thank you so much, Philip. Hello, everyone, and welcome to my today's presentation. And as Philip already introduced the topic, so in today's presentation, I'll be talking about lessons from the past, especially from past of machine learning, deep learning, success of deep learning, and then I'll talk about Imagenet Dataset. And after that, I'll be talking about deep learning bottlenecks. And then last but not the least, I'll be talking about 3 strategies to overcome data annotation bottleneck, and where I'll be discussing about transfer learning, active learning, and self-supervised learning. So there is so much to learn from the history of machine learning, and it has gone a lot of ups and downs, like seasonal changes. So here in the slide, when you see this sunny image, it represents spring season, and then fall, and then AI winter. So like seasonal changes, AI has also gone a lot of ups and downs. So it got very popular in 1960s, especially after the birth of multiple layer perceptron. And then it popularity picked up with the birth of another technique called backpropagation and development of convolution neural network. And after that, people start using these machine learning or convolution neural network on the smaller data set. And this is when machine learning got into a period of banishment, also known as AI winter. However, something very good happened in 2010. This very important thing was which made machine learning or deep learning very popular again. And this is the Imagenet Dataset released by Princeton University in 2010, and which resulted in the new revolution which deep learning has brought, and we are already seeing from past decade or so. So annotated dataset has a huge role in the success of machine learning. And Imagenet was the game changer, as I told you earlier, that it changed the game totally for computer vision experts. And in 2010, Fei-Fei Li at the Princeton University released Imagenet Dataset. And the beauty of this dataset is that this is a free data set with more than 14 million annotated images, with 20,000 categories, and with more than 1 million bounding box annotations for detection problems. So this all the images in Imagenet dataset is labeled by hand to indicate the objects that are pictured in these images and their corresponding categories. So in 2010, the Imagenet launched the large-scale visual recognition competition to see whose algorithm could best match the images to their categories. And this year, the year 2012 was a breakthrough year. And why I'm saying breakthrough, because this changed the application of artificial intelligence, and it was also breakthrough for Imagenet. So in 2012, LXNet, a deep convolution neural network architecture, achieved the top 5 classification error rate of 16% from previous best year of 26%. And in following years, we see that other people followed their approach, and this multi-layer architecture was adapted by many other, leading to lower the error rate. And in 2015, we see the best human level accuracy for classifying these Imagenet data set. And we see that the human classification error reached at 5%, but in the same year, we see that ResNet model was able to beat the human level classification error by achieving the lowest classification error rate of 3.6%. And in 2017, in the final year of this Imagenet competition, the top 5 classification error dropped to 2.3%, leading to end of this competition. So in 2015, when deep learning models were able to beat human classification error rate, we also started thinking about applying machine learning-based models in plant stress phenotyping. So in 2015, when I started thinking about using machine learning and plant stress phenotyping, during that time, there were a few major bottlenecks in plant phenotyping. The first bottleneck was big data collection. And during that time, we started figuring out use of UAVs and robots in the field. And another bottleneck was big data analysis. And for that, machine learning came to the rescue. So to handle that bottleneck. So with that regard, we also had, with that regard, the new bottleneck came in with use of supervised machine learning method was big data labeling. So this data annotation became another bottleneck, which is still a bottleneck in our AI applications. So in our review paper, Machine Learning for High Throughput Stress Phenotyping in Plants, we recommended the role of machine learning in data analysis, especially when it comes to big data, and how to automate the plant stress identification, classification, and quantification and prediction. So suppose we have a farmer or researcher who is interested to apply, who's interested to use UAV or robots to do identification of a stress or classification, whether he wants to know how much stress is in field, whether it's low stress, moderate stress, or it's completely plants are stressed out because of abiotic or abiotic stresses, or it could be when a farmer researcher is interested in quantification of stress, that how much severity of a disease is, or how much insect pest is there, so that they can do the mitigation decision, whether they need to spray insecticide or pesticide or fungicide. And the next, the last stage where machine learning can be used in plant stress phenotyping is prediction, where we are using historical disease data, and we are using weather data, we are using stress images to predict the stress, whether in future these conditions, the stress can come or not. So machine learning can be used in these four scenarios, which I call ICQP paradigm. So till now, a lot of machine learning applications we have tried in our group. And basically, we have used a supervised machine learning in these applications. So the first application where we have applied supervised machine learning was in case of soybean iron deficiency chlorosis detection, where the main idea was, can we detect soybean stress or abiotic stress iron deficiency chlorosis using iPhone? So how can machine learning be trained to rate these soybean plants in field on a scale of 1 to 5? So this work got published in Plant Methods, and we just were able to show that supervised machine learning can be used to train a smartphone app to rate iron deficiency chlorosis severity on a scale of 1 to 5. Another application where we use supervised machine learning was rare object detection in case of soybean cyst nematode. In this case, the main problem was that the soybean cyst nematode egg detection. So in order, soybean cyst nematode is a #1 pest of soybean in Iowa, and it every year it causes billions of dollars of loss. So here the main idea was that every year when entomologists go into field and collect these soil core samples, and then they clean these soil samples to see how much severity of these soybean cyst nematode is in soil. So they basically are preparing these slides to detect the soybean cyst nematode in these slides. So we used here supervised machine learning encoder approach, where we were able to show that using deep learning, we can not only identify these eggs, which are these 3 eggs, which are in this cluttery image where we have other soil particles, but also we can remove the clutter. So and creating an application where plant scientists can use directly this machine learning for rare object detection. Another application where we use supervised machine learning was in case of multiple stress detection. And I'll be talking about this example in the dataset and other slides. And the main idea here was, can we use deep convolution neural network to differentiate 8 different stresses? And the last example here, which I want to talk about, is a deep learning application where we used hyperspectral signatures to detect for the early stress detection. In this case, we wanted to see soybean charcoal rot disease, which comes inside the stem of a charcoal rot. And the breeder can't see from outside. So we have to cut soybean stems into half to see the disease progression. So in this case, we used hyperspectral camera and we trained it on a machine learning algorithm to not only detect 3 days before it can be visual to human eyes, but also we were able to classify into disease and healthy category. So with these applications, as we were applying machine learning, we are also facing a lot of challenges in successful application of deep learning, especially when we are looking for outcomes, the tangible outcomes for various applications in agriculture. So one of the biggest challenge right now we are facing is collection of large label data. So when I say large label data, especially I'm talking about training a machine learning for supervised machine learning algorithm where we need labeled data. So collection of this data is good. We are able to do that with UAVs and robot, but labeling or annotation is a challenge right now. Another challenge we are facing in this big data collection and analysis is noise in the data, especially when we are flying a UAV and we are collecting a lot of data from field. So suppose we are collecting images for sudden death syndrome, severity in field, but now we see that there is not only just one disease, there are other diseases in field. So we need to remove other stresses or noise in order to just concentrate on sudden death syndrome and field. Or do the severity of sudden death syndrome. So the next annotation or the data challenge we are facing right now, as I said, is data annotation. We are collecting a huge amount of data, several terabytes of data, but we are facing a challenge for annotating it, labeling it so that supervised machine learning can be used for training a robust model. In addition to these data challenges, we are also facing modeling challenges. And one of the modeling challenges, the class imbalance. So suppose we want to create an app to detect 20 stresses in soybean, but in nature, we find that not all stresses come at a time. So we will have, some classes will have more data, some classes will have less data. Or we are interested in doing the severity of some certain disease. And then for that disease, we will see some classes have more severity and some classes have less severity. So again, we are seeing this class imbalance, which is right now one of the modeling challenge. The last modeling challenge is model selection in itself. So we have right now, we can see we have tons of various models. So which model, which deep learning model or which deep learning architecture we need to train our model. So how to select those model is also a challenge right now. So when we talk about annotation, so annotation of large sample, as I said, is very essential for developing any robust machine learning model. And these annotation tasks involves drawing these bounding boxes, especially for localization. For example, in this case, the problem was, can we detect sorghum head in these images we obtained from UAV? Another annotation task could be drawing these body fitted profile for segmentation, where the idea is to mark these disease regions in various leaves. And the third kind of annotation is assigning classes for classification. For example, this is an iron deficiency chlorosis stress in soybean, where the breeder is interested to classify them on a scale of 1 to 5, where scale 1 is highly resistant and rating scale of 5 is susceptible. And these annotation tasks are generally performed by either domain experts or trained personnel. citizen scientist, or paid Oracle or Mechanical Turk. These annotations can be very expensive in terms of time, money, and generally samples for annotations are selected randomly. So now the question is how to overcome the data annotation bottleneck and supervised machine learning. So the one technique which can be used to overcome data annotation bottleneck is transfer learning. And transfer learning is a machine learning technique where the idea is to train a model on one task and repurpose it for a second or related task. So basically, transfer learning is an optimization technique, and which is allowing for a rapid and improved performance when modeling a second task, especially in this case, has a smaller data set. So you can imagine transfer learning as, so we have trained this deep neural network model to identify or classify soybean diseases. And we are interested to transfer this learning, what we have learned for classifying soybean diseases, to classify now mung bean diseases. But mung bean diseases have less data. We don't have that much resources. Breeder doesn't have that much resources, and he wants to use this pre-trained network, which has been trained on large soybean diseases, to train to identify mung bean diseases. So in transfer learning, basically, we can do is we can use the pre-trained model, which was trained to identify soybean diseases, and we are freezing these initial layers, which are identifying these features, and we are fine-tuning these last few layers so that it can now identify mung bean diseases rather than soybean diseases. So this is known as transfer learning. So this transfer learning we used in our PNAS paper, where we showed that using a deep neural network architecture, which can identify these 8 stresses in soybean, which included bacterial, fungal, nutrient deficiency, and chemical injury, we can use this pre-trained network to identify similar stresses in non-soybean species. For example, using transfer learning, we were able to identify iron deficiency chlorosis in case of vines, and also we were able to identify potassium deficiency in case of cucurbits. So now question is why we need expert for annotation tasks. So as we are moving into more complex problem, we need an expert for the annotation task. For example, for a sorghum plant population where all the heads are same color, then we don't need any expert to do that annotation task. Because any person who is not aware can do this task. However, if these heads are of varying color, then we might need an expert to annotate. So in plant science, we need an expert for the task of annotation, where a problem requires domain-specific knowledge, and where Mechanical Turk or Oracle can perform the job accurately, especially in problems where annotation requires correctly identifying plant disease or doing severity estimation. So in this picture, this is the same disease, but with different symptoms. So if an expert knows that this is the same disease showing various symptoms, different symptoms, but there could be images with noise in the data where we have the disease of interest, but we also have another disease which we are not training that disease for machine learning model on that disease. So these are the problems where we need an expert to annotate these images. So now question is how to smartly annotate data for modeling these supervised learning model, or how to learn from fewer labels, so that we not only reduce the number of annotators and need to annotate these large data set, but also less images for data annotation. So another technique which we are following right now in machine learning world is known as active learning. So here the idea is, so active learning is a subset of machine learning technique, in which learning algorithm uses an acquisition function. Basically, the idea is we use this acquisition function, which adaptively selects most informative samples from a pool of labeled and unlabeled data for a human expert or an oracle to label. And we use this method, active learning method based on uncertainty acquisition function, like least confidence entropy, deep Bayesian. And we also are using diversity methods for these acquisition function, especially core set. So basically, the idea is here, using the acquisition function, active learning is selecting best samples or the most diverse sample or most uncertain sample for the human expert to label. And these new label sample becomes the training data and evaluation, and goes in evaluation process. And and this process is iterative. So these samples become the part of training, and we are training an algorithm again. And then we are finding the new labels. And once these new labels are labeled, new samples are identified, these labels are added into training. And so we continuously, iteratively keep doing this process till we have achieved the desired accuracy, or till our budget, we ran out of our budget. So we used active learning technique to select samples to train a mobile net version 2 architecture to classify around 15,000 stress images into 8 different classes. And here in this graph, you have to notice that when we are using these uncertainty or diversity-based active learning methods, like least confidence, entropy, core set, debug, compared to randomly selected samples, you can see that with a random selection method is giving lesser accuracy than these uncertainty-based or diversity-based sampling method. So when we use active learning method or diversity-based sampling method, we are seeing that with around 4,000 samples, this is very less compared to our transfer learning method, where we were using around 16,000 images to train the supervised machine learning. So here with 4,000 samples, we are getting accuracy of around 81%. So this is huge, that now rather than training a whole model on all the images, we can selectively select these diverse and uncertain sample from each class to train our ML model. So we found that this active learning method is selecting selectively sampling the classes where the accuracy is lower. So here you can see when we are using random method, so random selection strategy is blindly picking new samples for annotation from various classes, and the distribution of the selected sample is uniform. But when we are using these active learning method, you see that active learning method is, this graph shows the selected samples, and here this graph shows the accuracy. So in this case, you can see when we are using these least confidence method, it is selecting samples from the least accurate classes. So class 0 is bacterial blight, and class 7 is bacterial pustule. So you can see the accuracy for these two classes is low. So our active learning method is selecting more samples from class 0, which is bacterial pustule, and more samples from bacterial blight. So this confirms that active learning is selectively selecting samples, so that with less number of samples, now we can train a robust model which has higher accuracy, like 84% in this case. And when we compare these active learning results with our previous supervised deep learning on the same data set for stress classification, we see that in supervised classification also, bacterial blight and pustule had lower accuracy. And we can see this from the confusion matrix, that the deep learning algorithm is getting confused between bacterial blight and pustule. For bacterial blight, we see that it is getting confused with 72 samples as bacterial pustule. And we see that with active learning, what active learning is trying to do is select more samples from the classes where the accuracy is less, and where even the breeder gets confused when he's classifying these two diseases, because these have very confounding and similar symptoms. So this gives very much confidence to us that active learning is working, and we can train a robust model with less number of samples. So another, currently the hottest topic right now in machine learning world is self-supervised learning. And this self-supervised learning is an unsupervised machine learning where a model is based on two tasks. So the first task involves the unsupervised pretest task, and the second task is the downstream task involving simple use of a linear classifier to fine-tune the transfer learning. So basically the idea is, if we are interested to classify these images into a dog and a cat images, we are not using labels in a pre-training process. This is completely unsupervised method for pre-training. Basically, so there are these pre-test tasks which can be like we are rotating an image. In this case, we are rotating a cat or dog image. We are not labeling it as a dog or a cat. We are just trying to rotate each image of a cat and a dog on a rotation of 0-degree angle, 90, 180, and 270. And then in pre-training process or step, the machine learning is trying to learn these various angles. And when it is trying to learn various rotations, it is trying to learn the underlying representation of these images. And this learned representation then are transferred to a next step where we are using less images of a cat or a dog and classify into final classes. So this, instead of labeling images as cat or dog, you can use you can rotate them and the train model to predict the rotation. And when we can generate a virtually unlimited training data from images we have. And once self-supervised model learn these representations from these millions of images, which we have created by annotating or rotating, we can use transfer learning to fine tune it on some supervised tasks like image classification. In this case, identify cat versus dog using very few examples. So this is how this self-supervised learning works. So we used this self-supervised technique to do our multi-stress classification. And so for this, we again use the same data set, 16,000 images. In this case, we used around 15,000 images for training, and 10% of the data was used for test. And in this case, the base model was ResNet 18, and feature dimension is 512. So we used, like active learning, we used 4 different methods. We wanted to see various self-supervised learning methods, how they are performing when it comes to soybean stress classification. So in this case, we use 7 different self-supervised training methods for soybean stress classification problem. And these 7 self-supervised method compares the embeddings of feature space between the image and its augmented version using different loss functions. So these different methods are doing various different pre-task methods to do self-supervised learning. And then later on, we are doing transfer learning to do the final classification. So these methods, we used self-supervised method, contrastive learning, where we use SimClar and MOPU. We use clustering method of self-supervised learning, where we use SWAV method. We used distillation method, BEOL and NNBEOL, and information maximization method, which is Barlow twins. So before I go to the next slide to show the results, I just wanted to show here that we did self-supervised learning where we used a linear classifier for fine tuning, where we are using the freezing the early layers in the network, and then changing the last layer with linear classifier for the transfer learning. And another method was a full model fine-tuning, where we are using, fine-tuning the model end-to-end to learn the initializations. So here you can see that we used, so I'll just give you one example, and this example is Barlow twins method, and Barlow twin method of self-supervised learning, and another self-supervised learning method is VIC regularization. And we are comparing them with Imagenet and supervised learning method as a base method, because we need to know how well they work when we compared it with Imagenet initialized weight versus supervised machine learning. So when we are doing the self-supervised learning, where we are doing linear model fine-tuning, we find that the self-supervised learning method, or the Barlow twins, is working better than Imagenet, which is here, and supervised machine learning. And we see that especially when we have less number of samples, we see that our self-supervised learning method is giving more accuracy, around 93-94% compared to self-supervised with around 10% of the samples. And in full model tuning, we are seeing, again, these self-supervised methods are working better than Imagenet and supervised models. And in this case, we see that Imagenet is, the model is initialized with using Imagenet weights, and it is, the end-to-end training has been done, and it is working somewhat similar to our self-supervised learning. So this gives us a lot of confidence that with 1% of the training data, so Barlow twins with just 149% images is giving us dataset accuracy of around 83%, which is much higher than active learning, where same accuracy was obtained using 4,000 samples, or compared to supervised learning, where we were using all the data, around 16,000 images, and we were getting around 94% accuracy. You see here, when we are using 5% of the data, we are getting 92% accuracy. So with less number of samples, we are getting higher accuracy when we are using the self-supervised learning compared to Imagenet or supervised learning. Similar results as I show in graph, again, when we are doing self-supervised learning with full model fine-tuning, again, these self-supervised methods are winner. And in this case, Barlow twin is again the best method. And we can see with 3% of the data, we are getting 92% accuracy, which is similar to what we are getting with self-supervised model, but we are using all the data, 16,000 images, but here we are just using 3% of the data. So with this, so we are also trying to utilize self-supervised learning and our insect detection and classification problem. And we are seeing similar results as we are seeing for disease problem. And the best part is in insect classification problem, our data is very imbalanced. So the class imbalance is huge, but we are seeing that self-supervised based learning not only trains model with less samples, but also it is able to handle class imbalance. And so takeaways from self-supervised application and stress phenotyping, we found that Barlow twins was the best self-supervised method for annotation efficiency. All the 7 SSL methods perform significantly better than the supervised baseline models for linear and end-to-end evaluation. And we also observed that SSL methods were robust to class imbalance and highly efficient for soybean stress classification. And currently we are working to identify the best combination of self-supervised and active learning methods for annotation efficient image classification, segmentation, and object detection applications. And here in short, I would like to just show that how we started this work in 2014, and by formulating these various problems where machine learning application can be used. As I showed you that initially we have these problems where we thought will be best, because not all problems are there in agriculture where we can apply machine learning. So we have to choose problems where the machine learning application will give us a maximum output or application. So we started in phase 1 with formulation of problems, and we evaluated and selected various solutions by working on these problems. And in next phase, we are already moving from this phase 3, where we have already implemented some of these problems at a small scale implementation. And right now in research, we are trying to go at a production scale or research scale, where we are trying to implement these solutions on a large scale, so that soon we can go in phase 3, where we can do commercial application of these ML techniques for creating various tangible outputs. And when we started with this idea that in future we can use UAVs and robots by providing artificial intelligence. And currently we are in our program, we are trying to use these tools where we are using UAVs, robots to take these data, to collect the data on various stresses and agronomic traits and yield, and create some tangible outputs using this deep learning or machine learning tools and techniques. And this all work has been possible because we changed the way how we train students. So in my group, we tried to train our PhD students in plant science on engineering problem, where they're trying to work with engineers to bring machine learning and solve their plant science problem. And I also co-guide these engineering students who work with me on a plant science problem to apply these cool ML techniques, so that we can create something which is useful for the farmer, for the industry, and for the research community. And this year, our team at ISU received 2 big grants. So in AI and agriculture to continue our work. And the first grant is QLS, which is a Frontiers in CPS grant, where we are working in closing the loop problems using various sensing modalities and modeling and actuation and agriculture production related problems, again, to create these tangible outcomes in the form of tools for farmers and researchers. And the second grant is AI Institute grant for resilient agriculture, where we are working on crop breeding and production scale problems for resilient agriculture. So with this, I would like to acknowledge my collaborators, Shamik Sarkar, Bhaskar Ganapati Subramaniam, who are ML expert and computing expert, Dr. Arti Singh, who's a soybean breeder, who works with large data sets. Kaushik, I want to special thanks to Kaushik because he's a very diligent student and has been working for past several years with us. And he's the one who worked on active learning and self-supervised learning problem. And my PhD student and Shamik Sarkar's past PhD student who worked on these problems. And with this, I would like to take any questions if you have.
Thank you very much, Arti. That was a very, very informative overview about, for first, the history of machine learning applications, and then, of course, also a few of the problems, the bottleneck that you mentioned, the annotation bottleneck, and also means to overcome this bottleneck via self-supervised learning methods. So one of the questions that struck me while you were presenting was, is there even a need for manual annotation if those self-supervised learning methods do perform all so well? So you mentioned, of course, during your talk that there are special cases where the algorithm gets unsure, yeah, when you, for example, have a mix of several diseases or a very high noise in the image. However, you also showed that some of these self-supervised learning methods do outcompete manual annotations. So do you foresee, maybe with a perspective to the future, that manual annotation will become obsolete by time?
So this is where we are trying to go, Philip, that how can we reduce this manual annotation. So currently, like I said, active learning, where we are trying, earlier we thought that human in the loop is good to make sure that we are annotating things properly and it is going through it. But again, human annotation, as I said, it needed 4,000 samples still. 4,000 is still a huge. So that then came the concept of self-supervised, and that's where we are very much interested, where algorithm is itself learning the annotations and it's transferring it in the last stage to the final classes. So I foresee that in future we will completely try to go away from manual to automatic annotation. So this is where the future is. And we might be doing some very small scale annotation for the ground truthing or training based on like when for specific cases where the problem is very complex, where we can start with some manual annotation and train the model by those specific annotations to maybe annotate larger data set. So yeah, I foresee that for sure.
Mm-hmm. Okay, then we have one question coming from the audience from Mortiza Almazia, and I will try to provide her the option to send this question into the chat. Otherwise, we will try to promote her into the panel so that she can ask or he can ask the question themselves. So Mortiza, please write your question into the chat if possible. If not, we'll try to figure it out differently. For the meantime, I have a different questions. So in plant phenotyping, large quantities of image data are being produced at a very high rate. That's what you also mentioned in your talk. However, many of this data is unannotated. And you already explained that there are means to overcome this and how these means do perform in comparison and where the future goes. But there are also community-driven projects such as, for example, MyRP that target at annotating data sets born from plant phenotyping systems or plant phenotyping experiments. Do you think that these community-driven projects that produce a minimum information criteria for annotating those data sets, do they prove useful in context of AI? Or is that more of use in other fields, but not specifically targeted towards AI application?
Yeah, for sure. I guess citizen science is playing a leading role, and this is how I think Imagenet came into being. So they use citizen science to manually annotate, to create that data set to prove, hey, we need big data, we need annotated data. Mm-hmm. So again, as I said, that creating, using citizen science, I think till we are able to go where these machine learning can automatically annotate, we will be depending on citizen science. And I think it's not just, I think like plant science is a very complex discipline. And when we work on these problems in field, it becomes much complicated, because in field, we are not, especially for plant stress phenotyping, we are not just seeing one stress, we are seeing these stresses with confounding symptoms. So, and there I see that we need experts. But I think till we move to that automated annotation, I think we will be needing these citizen scientists or mechanical Turks or Amazon Turkers to annotate our data to make it more robust. And I will say, if at the end of the day, if we are commercializing a product, it depends how much money we are spending and what we are getting output. That's where these all annotations started. So we have to see whether it's a public tool we are creating or it is for the commercial purpose. So, and there is also what you call competition to release these things faster. So what will give you faster results, that also dictates. And if you are okay creating a tool and you think that, hey, citizen scientists can take a year and annotate for you, go for it. But if you are commercializing a product, then you don't want to wait for a year. You want to annotate at a faster rate. So it depends on which problem, complexity of the problem, end goal of where you're moving. And yeah, so it's again, a lot of factors which goes into it.
Yeah, in the case of MyRP, of course, the ones annotating the data sets are the researchers who are performing the experiment. And then, of course, they are somewhat of experts in regards of this particular experiment. So the quality and the timeliness of this annotation is certainly very, very robust. So, of course, basically, you're saying more annotation is always helpful when it comes to digestibility, or is there particular aspects you would stress being annotated in, for example, a disease experiment?
Yeah, it depends. Yeah, annotation depends what your problem you are towards. So if your problem is it depends on the problem. So come again with your question. I think I missed your question somewhere. What was your question?
Yeah, the question was, does that provide, or what proves most useful when it comes to annotation if you consider data set being annotated by the expert that did the experiment? So what is the best annotation you can get?
Yeah, it depends, first of all, which model you're using. You're using supervised machine learning. So supervised means annotation. This is where the success happened, where we are using supervised models and we need a lot of annotation label data. But if you're using unsupervised or semi-supervised, then we can bring expert in the loop, like active learning, which is a semi-supervised method where we are trying to bring human in the loop, where we also know that annotation is very tricky. So we can just get away with some small sample, the diverse sample which our algorithm is selecting, and making them to go with the expert. Because, and this I feel we are seeing in our day-to-day plant stress problems. So one problem which we are currently working, so some people think that this is the same stress, and I say, no, this is not the same stress, but we have collected images for one stress, but it is a mixture of stress. So if I give it to an annotator, they don't know what are the symptoms of various stress, so they will just say it is one disease. But when it comes to me, I say, no, I knew that in that year, we had maybe abiotic stress, we had 2 diseases in field. So then those samples, and if it is a supervised learning model, I need to make sure what we are training our algorithm is getting trained on that trait, not it is getting a mess. So this is what it's true for all supervised machine learning. If you are feeding garbage in, you will get garbage out. Then you can see that we have trained it for a particular disease, and it is not identifying that particular disease because we have confounding symptoms. And it is very tricky in plant stress world because all these stresses have so much confounding symptoms, or when it comes to insect detection problem, again, these insects can be so similar. So sometimes ladybird beetle, there are so much variability within that lady, and there are spotted lady beetle which are good and some which are bad. So how to differentiate, so we need expert when we are seeing that stresses. So yeah.
That was exactly more or less the answer to our first question, which came in through the chat from Farshad. And it was asked, dear Arti, thank you very much indeed for your excellent presentation. Is it possible to use active and supervised learning for plant phenotyping on combination of diseases and pests?
Yeah, I gave you 2 examples where we are using active learning, and now we are finding that self-supervised learning is working better than active learning. With 149 images, we are able to get 84%, but with the active learning, we need 4,000 samples. And another thing what we are learning from these self-supervised method is that earlier, like if it depends, like you are, as I said, model challenges also. So you want to use, you're fine-tuning your model, or you are using end-to-end model. When you are using fine-tuning, that means you are using a model as it is and just fine-tuning at the last layer. So, and we showed in our research that self-supervised learning, which has in-domain data set, works better compared to Imagenet, which has representations not exactly from stresses, But when we are doing end-to-end fine-tuning, we found that in-domain self-supervised learning works similar to Imagenet, because we are initializing the model using Imagenet. So we need to know in which scenario in-domain data set will be useful, in which scenarios out-of-domain data set will be used, so that we can train a robust model. Because right now, the machine learning world is flooded with these various architecture. Every day the paper is coming. So being a plant scientist, we need to know which architecture and why we are using it exactly at the end of the day. Like in QTL mapping world, we knew that, hey, if we have around 200 lines, we get, or more than 100 lines, we get better QTL estimate. But right now in plant phenotyping, what we don't have this number that, hey, for each, if you are using this model, exactly you need 100 samples from each class. We don't have. So this is where we are trying to standardize approach, that if you have a problem like this, maybe you don't need to collect millions of data set. With maybe 200 images, you can still get more than 95% accuracy. So this is where we need to go, and this is where we are trying to go, so that we can be certain that with how much data needs to be collected, which model we are working. So it is data-centric approach or it's a model-centric approach. So this is where we are leading.
Is this then also different between or specific for each different species and each different disease type? The numbers of data points you will need for robust output, or is it more like image quality or the way that you have obtained these images that plays more important role in this regard?
Yeah, so like with our plant disease and insect where we are applying, we came to know that even we have imbalanced data set, so like frog eye leaf spot has 1,000 data point, and healthy and other diseases have 2,000 or 4,000. Some self-supervised learning is able to predict both the classes better, even with class imbalance. But when it comes to active learning, maybe it not accounts for imbalanced class, So it depends also which method you're using. So these questions are very important, especially when you are trying to bring out an application, I would say, when we are just publishing a paper and they're telling, hey, this much, that much accuracy. But at the end of the day, if we are creating an app for maybe classification of diseases, a farmer is using, as soon as our algorithm predicts wrong, the trust goes down. So it depends what we are trying to achieve. At the end of the day, if any commercial product goes out, if it has accuracy, like maybe it can accurately identify 100 diseases, and with the 97% accuracy, that is acceptable maybe. So that research also we are trying to do in our new projects where privacy and trust issue, how can we target those issues when it comes to gaining trust with any application or tool.
Yes. Okay, thank you. Thank you for the great presentation. Is our next question. What's your suggestion about using machine learning-based techniques for extracting stomatal details? What will be the best code-free environment as for me to work on it? Do you have any?
That's a very tough question. I think it depends what's your objective, stomatal details. So yeah. So if you're talking about image-based phenotyping, you have to see how much variability you have in the stomatal trait you are targeting. So if your samples are covering all the diversity, then we are good. We don't need millions of images if you think 100 samples is covering all the diversity. And then we can use these 100 images, and when we can use transfer learning, we can use already pre-trained model and transfer, do end-to-end tuning, and get a very good model which is able to identify or predict your trait. It depends on your trait complexity and variability you have, and how to deal with it.
Oh, and it was added that it's specifically interested in data extraction of stomatal traits from microscopic images of stomatal imprints. So does it play a role, the scale of the image in digestibility?
Yeah, scale will play a role. So whenever we are training, we try to scale them at some resolutions where you are able to definitely differentiate your trait. So I think scale in resolution and scale in number will depend. And as I said, if it is able to cover the variability you have in that per trait, you are good. And even with a smaller data set, you can train a robust model by using your transfer learning or a supervised learning.
Next question. For self-supervised learning, you seem to still need large man-made annotated datasets. Can self-supervised learning systems not find relevant data themselves on the internet and derive their annotation by iterative learning processes?
No, I think you are a little wrong when it comes to self-supervised. That's the name where it is supervising annotation itself. It is trying to, as I said, if you have to differentiate cat or a dog, in an initial stage, it is either trying to rotate that image of cat into various angles and trying to predict the angle. When it is trying to predict the angle, it is also learning that this is a cat. So whatever underlying representation it has learned, then in next stage, we are using transfer learning, where we are using small, very small data set where the expert can come that, hey, this is a cat. Maybe with 10 samples, now you can classify thousands of images of cat and dog. So yeah, you need supervision, but at the last stage with very less images. As you said in earlier question, that we will need some human annotation. Yeah, we might need some annotation in final stages to transfer it to final classes. So yeah, that's true.
Next question. Thank you for the great presentation. What's your suggestion about using machine learning based techniques for extracting? Oh yeah, that's again the stomatal details. Do you have any recommendation for him on what software packages to choose when he or she wants to try to extract or digest images of stomatal details? Is there anything free that you would suggest, maybe to try a little bit and get into the whole topic?
So yeah, I think it will be better if you connect with me at my Iowa State account and send me some pictures and what is your scale of the data size or the resolution, and then maybe we can get back to you like how to approach that problem. I think that will be the best scenario for me, because I have never worked on stomatal problem, that trait. So I think I would like to see, and then maybe we can suggest something. So please use my Iowa State account. You just Google my name, and at Iowa State website, you can find my email ID.
Yeah, a link will be also available on the Phenomics webinars website of the webinar today. So easy to get in contact with Arti. If you run into problems, you can also send me an email. So there was one last question. I don't know exactly to what it refers, but Alexis Carlier did ask, is it also working for regression problem?
Yeah, I think right now we are just targeting classification problems. So I'm sure it should work on regression problem as well.
Okay. All right, Arti. I run out of questions, which is a good thing considering we have our time spent. It was a great pleasure to have you here, Arti. Where can we see you in the near future? Will you be attending any conferences? Will you be present at the North American Plant Phenotyping Network annual meeting later next month?
Yeah, I will try. I have not yet registered, but yeah, I plan to give you presentations here at in US, and we have NAPB meeting coming, and where we really want our IPPN and NAPPN community should attend that presentation, because it's a National Association of Plant Breeders meeting, and we have excellent line of speakers, and we have a session on prediction and machine learning. And I am chairing some sessions, and I don't know that I'm speaking in that session or not, but surely we would like you all to participate and attend that meeting.
So look out for the ASPB meeting.
NAPB.
NAPB, sorry.
Yeah, I'll send you a link for NAPB meeting. It's in August.
In August, NAPB meeting in August. Be sure to follow the session on machine learning and AI.
And predicting the unknown, where we will be talking about how in modeling and machine learning are combining, and genomic selection. So they are very interesting topic there.
Very well. We'll certainly have a look out for this. If you want to watch a recording of today's webinar, like for any other Phenomics webinars, you will find it right on our IPPN YouTube channel. Also, Arti, you will be listed there in the near future once I've uploaded this video. So check in there tomorrow or the day after tomorrow. You will be able to see the recent and all the past webinars of 2020 to 2022. Stay tuned until our next webinar, and follow our Twitter and LinkedIn channels for updates on when the next webinar is. I thank everybody in the audience, and I especially thank you, Arti, for speaking and educating us today. Have a fantastic rest of the day, and we'll stay in contact for the future. Thank you very much.
Yeah. Thank you, Philip, and thank you everyone for attending today's presentation. It was fun answering questions and giving a talk. Thank you. Bye-bye.
Bye-bye.